Hello everyone. Welcome to online course Microbial Toxicology Module 2. Module 2 is about bacterial toxins affecting generalized tissue. It is diphtheria toxin and its mode of action. As we have discussed earlier, exotoxins are various kinds. Exotoxin affecting generalized tissue. Diphtheria toxin is one of the example that comes under this category. And AB exotoxins affecting specific host sites. Neurotoxin, tetanus and botulinum toxins, and enterotoxin, cholera toxin comes under this category. And membrane disrupting exotoxins, again there are two types: protein membrane disrupting exotoxins, enzyme membrane disrupting exotoxins, and super antigen exotoxin. Staphylococcus enterotoxin can be considered as super antigen exotoxins. Today in this module, mainly we are focusing on AB exotoxins affecting generalized tissue, that is the diphtheria toxin. Diphtheria toxin produced by Corynebacterium diphtheriae and was isolated by Raux and Ersin in 1888 and is an important virulence factor in the pathogenesis of diphtheria. Rats and mice are relatively resistant to diphtheria toxin, but human beings, rabbits, guinea pigs, and bird cells are very sensitive to diphtheria toxin. And diphtheria toxin is made up of its two subunits A and B. And A subunit is very important, and it is mainly participate in catalytic action, and toxic effect is responsible by the subunit A, and its molecular weight is 21 kilodaltons. And the large subunit B, main function is which attached to the host cell membrane, and its molecular weight is 37 kilodaltons. Mode of action of diphtheria toxin. Diphtheria toxin mainly affects the elongation factor of the host cell. Elongation factor is one of the important molecule essential for the translation mechanism or protein synthesis of the host cell. Diphtheria toxin mainly damage the elongation factor so that the protein synthesis of the host cell will be ceased, and it leads to the death of the host cell. This is the main action of diphtheria toxin. And we can see in this picture, diphtheria toxin is AB exotoxin, and the A subunit is mainly responsible for the toxicity. And when the B subunit attached to the host cell membrane toxin receptor, the A will enter into the host cell, and A is engulfed by the host cell membrane, and endosome formation will be takes place, and this endosome acidifies and A subunits will be released into the cytoplasm of the host cell. Here, the A subunit transfers the adenosyl group from NAD, nicotinamide adenine dinucleotide. From this molecule, it transfers the adenosyl group to EF2, elongation factor 2. And so because of that, EF2 lost their function, and the protein synthesis will be ceased out, and it leads to the cell death. This is the working mechanism of diphtheria toxin. And in this picture, you can clearly see that this is the A and B subunits of the diphtheria toxin, and B subunits attached to the toxin receptor and the host cell membrane. And this, because of the attachment of the B subunit on the host cell membrane, the pore formation will be takes place on the surface of host cell membrane. Then the A subunit slowly travels through the pore and it reaches to the cytoplasm. And here, this A subunit mainly attached to the elongation factor 2, very essential for the protein synthesis, and it modifies the elongation factor 2 by attaching the adenosyl group from NAD. And this modified EF2 will not participate in the protein synthesis, so that the protein synthesis is shut down. It leads to the death of the host cell. This is about diphtheria toxin and its working mechanism.